All right. Well, good morning, everybody, or good afternoon, good evening, depending on where you are. Welcome to the Variant Interpretation for Cancer Consortium Virtual Molecular Tumor Board. Today, we have guests from Constantium Biosciences. We have Nicholas Schafer, Daniel Goldman, and Norman Ong, who are co-founders of this startup company that is focused on using functional evidence for clinical variant interpretation. So Daniel reached out to me a few weeks ago, and I was really glad we were able to fit you into the schedule. So really interested to hear what great work you've been doing and to kind of spread the word about gathering functional evidence. So thank you so much for being here. So without further ado, I think Nick is our primary presenter. So go ahead and get started.
Yeah, great. Thank you, Beth. And thanks for everybody for coming and for the opportunity to speak here today. And as Beth mentioned at the top, please feel free to jump in, and then we'll also have some discussion at the end. So the title of our presentation is MAVEvidence, a functional evidence platform for clinical variant interpretation. I am here with my two co-founders of Constantium Biosciences, Daniel Goldman and Norman Ong. Fortunately, we are not alone in this venture. We have support from the NHGRI in the form of an SBIR grant that I'll talk a little bit more about later, and also a great set of advisors. So Fritz Roth is our founding scientific advisor and a pioneer in the kind of high throughput functional assays that we'll be talking a lot about today. VICC and Brian are physician scientists with specialties in cardiology and cancer, respectively, and sort of also pioneers in their own way in doing research in this area, but also applying this kind of data in the clinic. And Jean and Eric are assisting us on the product and business sides, respectively. This is a picture of that same group of people from our recent all-company meeting. Everybody's looking pretty happy there. And before we jump in and tell you about the platform itself, I thought it'd be helpful maybe to give you a little bit more background about the company, how it came to be and what we're about. So the company has really started, with the motivation that would be familiar to a lot of people on this call, out of our desire to, use our skills to help patients. And for me, it was like a series of events that brought me to it. I had done a PhD that was focused on computational methods for studying protein folding, and then a postdoc where I was making new experimental assays for studying mutational effects in membrane proteins that leveraged a lot of automation to help me get that done. And then I had gone on to write a faculty research proposal where I was proposing to combine experimental and computational methods to probe mutational effects in proteins that are involved in neurodegeneration. Now, in the end, I chose to prioritize staying here in Houston, which is where I'm located. And so that forced me to broaden the scope of my job search. And I ended up as the director of data science in a company here in Houston in a completely different industry. And then, very quickly, I experienced several kind of events that caused me to question this decision a little bit. First of all, that was right around the time of the pandemic, and the need for more and better practical applications of biological research really became painfully acute for all of us. And also right around that time, I was in the hospital for the birth of my daughter when a nurse tipped me off to the fact that my son, who was 3 at the time, was showing signs of autism. And we took him to a neurologist, and he was subsequently diagnosed. And me being the person that I am with the interests that I have, I had a whole genome sequencing done for 8 of my closest relatives, including my 2 kids, and found out that, oh my gosh, my kids have an NF1 variant. Now, because I had, so many relative sequence, I found out that I also carry the same variant, and my unaffected mother also has the same variant. So that was sort of my first personal run-in with variant interpretation. And then also right around the same time, my father was diagnosed first with Parkinsonism, and later that diagnosis was revised to Alzheimer's. And because I already had the genome data available, of course, I went looking and found that I share with him a likely pathogenic variant in the GBA protein. And I took this information to a clinical trial that was enrolling both healthy and people with Parkinson's symptoms, and was ultimately denied admission into the trial on the basis of my age. But all of this stuff came together to get me kind of thinking about, like, how can I apply my skills better to help, people who are in situations like my family. And I know that, like, every one of our founders has some kind of similar story, as do probably a lot of you. So I got together with these guys, and we formed our team, and for about 2 years we were working nights and weekends to identify a specific problem to work on, come up with a plan for the company, and then apply for the grant, which we were ultimately awarded late last year. So we have a phase 1 SBIR grant, and we've also raised some additional funds from family and friends, and those two things combined have allowed us to start full-time work either late last year or early this year, depending on the founder. One of the very nice things that the SBIR program provides is a supplement called the iCore program. And so in the last several months, we participated in this intensive customer discovery program called iCore, where the goal is, in the span of 2 months, to interview 100 people who are stakeholders, in our case, in the field of genetic variant interpretation, to learn about what challenges they're facing. And currently, we're in the process of developing a commercialization plan in addition to a research plan for our phase 2 application. And the commercialization plan needs to demonstrate that we'll be able to reach self-sustaining revenues and/or attract private investment after the phase 2, which is an NIH requirement for the phase 2 grant. Okay, let's talk a little bit about how we see the problem of variant interpretation currently. So I'm going to walk through a couple of lines of evidence and talk about sort of the opportunities and the challenges as we see them. So we have population data, which obviously requires sequencing a lot of humans and being able to safely and responsibly share that data. And there are, of course, some excellent projects in the works, including the UK Biobank. Still, this is a slow way if you want to observe the impact of all possible variants. Likewise, with segregation data, there's currently no good solution for both obtaining and sharing this data at scale, but we do think it could be an opportunity to create a scalable resource, although it's not where we're focused at the moment. And it does have some of the limitations that population data has. When people think about scale, they often cite computational predictors as a way of scaling, and there's some truth in that. Of course, ultimately, if you keep digging through the literature, what you'll find is that inevitably the computational predictors are themselves based on experimental data, and therefore ultimately not a truly independent line of evidence, although there will be a lot of value to variant interpretation that comes from applying models to different kinds of experimental evidence, which currently goes under the guise of computational predictors. And finally, there's where we are currently focused, which is functional data that's collected in the lab, where right now technological breakthroughs are driving a massive increase in the velocity at which we're obtaining this kind of data. And what we found is that we need ways to facilitate the application of this data in practice. In particular, we see a couple of factors coming together here, creating a sea change. So there are new gene editing technologies and the advent of these massively multiplex experiments that we'll discuss, such that over the next 5 years, we expect the research community will perform hundreds of these experiments, generate functional data on millions of variants across thousands of genes. And if fully leveraged, this could really be a key to solving the variant interpretation problem. how does this fit into the kind of wider scope? On the left, we have a sort of schematic representation of how we see the present and the future of these fields and how they're related to each other. So precision medicine is a kind of common buzzword, and it's a worthy goal. And genetic testing currently partially underlies that, and we see the influence of genetic testing on precision medicine growing over time. Of course, with genetic testing, we need, as we discussed, good methods for variant interpretation, and that's where we see functional genomics playing an increasingly important role in covering the large number of possible variants. And just to sort of give a taste of what the future holds, on the right, we're highlighting an abstract from a recent paper where they highlight having probed the functional effects of 52,000 ClinVar variants across 3,500 genes in this single study. Now, these base editing techniques currently, they don't yield the kind of same quality and level of data that individual gene-focused high-throughput studies focus, but these technologies are advancing, and sort of this is kind of what we see coming down the road. So that brings us to what MAVEvidence is, which was in the title of the presentation. And as we mentioned, it's a functional evidence platform to realize the promise of functional data for variant interpretation. So we have these hundreds of experiments being done across thousands of genes, generating data on millions of variants. And the idea is to take all of that in, perform the necessary data analysis, validation of the assays, standardization of the information, and calibration of the evidence. Because as everyone on this call is aware, properly calibrating evidence is a key to responsible variant interpretation. That in turn can help to drive variant classification for the purpose of precision treatment, diagnosis, and further research efforts. Okay, that sounds great, but how do we actually accomplish this in practice? The platform needs to be both scalable and sustainable if we're going to keep up with this flood of data. And this is just a kind of sketch of what the workflow might look like. So on the left, we have several different kinds of data coming into the platform. One, we first need to identify and prioritize the functional studies that are being published in the literature, and then extract and harmonize that data into a functional data database. On the bottom, you have the no less important clinical data, and that needs to be extracted and harmonized and gathered into a clinical database. And the functional data itself, so I mentioned that there are various kinds of computational methods that can be applied to help refine and model the data, but ultimately it needs to be combined with the clinical data so that those assays can be evaluated and calibrated to turn that data into functional evidence. Now, once you have the functional evidence sitting there, it's of course not doing anyone any good unless it's being used in practice. And so you need APIs and applications that are well maintained, as well as sales and support people to reach the customers and support them in their application of the functional evidence. So what are we hoping to accomplish by doing all of this? Basically, it's a paradigm shift from so-called reactive functional testing, where you first observe a variant, and then maybe later you do an experiment on it and get some information about that variant that can be applied for future patients. Here, with the advent of these high throughput experiments, what we're hoping to do is to do proactive functional testing. So you identify one or more genes of interest, you probe many, many variants within those genes, possibly to the level of saturation, and therefore hopefully obtain many more meaningful classifications, leading to fewer uncertain variants, accelerated diagnosis, and ultimately better patient outcomes. So I'll talk a little bit about the I-Core interviews that we did. We tried to get a wide variety of stakeholders in our interview process across the industry. And this shows a breakdown of the first 80 interviews that we did by job type. So we talked to many clinical geneticists and lab directors, in addition to genetic counselors and bioinformatics scientists and physicians. And in terms of where these people were located, not surprisingly, we got many people from academic settings that were wanting or willing to speak with us, in addition to many people from both more general diagnostic companies and specialized genetic testing companies. So what did we learn during I-Core and also since then? First of all, the good news is that there is a strong desire to leverage this kind of data as evidence both in commercial settings and non-commercial settings. In practice, currently, because of the relative novelty of this kind of data, the level of usage varies wildly, as do the methods for applying this data as evidence. So there's kind of a lack of standards currently in terms of how to apply the data as evidence. And one kind of external factor that's changing things a lot currently is that commercial labs in particular have been hit very hard in the last few years. And with some of them laying off as much as 50% of their employees in an effort to cut costs. So what that means is that the current environment is not that conducive to spending money on implementing new systems within the company to ensure that all this kind of new evidence is fully leveraged. On the other hand, we did find during our interviews that these companies are already spending a lot of money in the form of variant scientists' time on evaluating functional studies for variant interpretation. So people recognize that it's an important data source, and they care about both saving time and maintaining the quality of the interpretations that they're doing. And so, especially since I-Core, we've been doing a lot of work to understand what are their current systems and needs, and how can we build something that will actually get used in practice. So we're actually going to switch gears now to talk a little bit about variant interpretation frameworks, but I wondered if anybody had any kind of questions or comments from the discussion so far. If not, we can also talk more at the end.
Sure, you might have answered this already or plan to, but how do you deal with like tissue type specificity? So you're looking at a variant within a gene. Maybe that gene operates differently or is expressed differently in different tissue types. How do you deal with that? Are your functional studies very, very focused on a specific like tissue type?
Yeah, I think that's good. So we're going to talk a little bit more kind of about what the platform does. And so I think it'll be a good opportunity to discuss stuff like that. Oh, somebody raised their hand.
Yes, Manuela. Oh.
Manuela, we can't hear you.
Yeah, sorry, my Zoom was really slow. Yeah, I was just wondering, so thanks for the intro. How did you decide on the type of data you actually put into your platform? So MAVEDB, for example, but there are probably others out there which you could use. And so why do you focus on those?
Yeah, so I'd say that the focus on high throughput functional studies is there's a couple of reasons for that. One is that, it makes obtaining a lot of data easier. And also these studies have the advantage of having basically internal controls and covering a lot of variants that allow you to sort of evaluate their quality. In terms of, which high throughput functional studies to look at, we're working hard to develop a system that allows us to basically evaluate in an objective way, all kinds of data that comes out. And so, we don't want to be picking and choosing ahead of time which studies we decide to evaluate, but doing the rigorous evaluation, informs how to apply that kind of data. And you'll see a little bit more later when we show some screenshots of the platform, kind of the approach we're taking to that a little bit. So yeah, so in terms of we were really excited to see this recent paper. Obviously, this group is a major contributor to this framework, and having been working mostly in the germline space, or thinking mostly in the germline space, we're really excited to see not only are there standards being developed on the somatic side, but also that functional data, like in the case of germline interpretation, has sort of a place. And we really value these kinds of standards because it gives us a place to start from and work within in applying this kind of data to variant interpretation more broadly. And so, I don't need to explain to this group that functional data has a place in these in this oncogenicity framework, and it sort of largely mirrors its place in the corresponding pathogenicity framework from the ACMG. And in terms of the types of studies, as Beth and Manuela were pointing out, they're quite heterogeneous, but the kinds of studies that we're thinking about follow this basic pattern that's illustrated on this slide. And so, typically you have some population of cells, you make a variant library and introduce the variant library into the cells. And then, one of the key driving technologies is sequencing. So you typically sequence at the beginning after you've introduced the variants into the cells, and then you apply some kind of sorting or selection, and you sequence again, and possibly over multiple time points. This sequencing data is then transformed into so-called functional scores, typically one for each variant that's studied in the assay. And because these are high throughput methods, you can cover thousands to tens of thousands of variants in a single experiment. One of the advantages of that is that some subset of those variants will have existing clinical annotations. And as is sort of illustrated by the histograms on the right, you can do an inspection of how good of a job does this assay do in separating known clinical annotations, pathogenic and benign, for example. And you're collecting data on thousands of variants, many of which will not have been seen before in the clinic, but that we know if we sequence enough people, we will eventually see all of these variants in humans. And there is already a move to put this data into practice. So here I'm highlighting a study where MAVEs for BRCA1, TP53, and PTEN were evaluated by folks at Ambry and University of Washington for their ability to reclassify variants. And their finding was that they took all of the VUS in addition to the types of evidence that were used to classify those variants initially. They added to it whatever functional evidence was warranted by the strength of the assay, and found that as many as 50% of VUS for BRCA1, 69% for TP53, and 15% for PTEN could be reclassified out of the VUS category. And as we've suggested, we see a sort of coming flood of this type of data, and there are already studies for BRCA1, BRCA2, a whole bunch of cancer predisposition genes. The pace is accelerating. We fully expect there to be many more assays to be published just this year. And there are many challenges that variant scientists face to responsibly evaluate and apply this kind of functional data. So this is where MAVEvidence comes in, and the basic workflow is illustrated here. We have raw data coming from multiple MAVE experiments in general for a gene. That data needs to be mapped onto the sequence, and then using the clinical data, we perform an assay level validation, but also a variant level calibration of the appropriate strength of the evidence, and then build an application to make all of that data searchable and easily obtainable by a variant scientist who's in the process of putting together lots of different lines of evidence to perform a variant classification. these evidence could be calibrated using customized reference sets. So right now, we have the notion of pathogenicity in mind, but there's also the separate but related notion of oncogenicity that could be used to calibrate the data. And the platform currently has about 120,000 pieces of evidence that cover 80,000 variants in 12 different genes. And our goal is to be able to keep up with the literature. So as soon as a assay is published, we evaluate that assay and make a decision about whether or not it's appropriate to include it in the platform. The evidence is calibrated using, again, like published recommendations. So within a Bayesian framework that was an elaboration of the ACMG scheme. So calibration depends both on the performance of the assay overall and the score of the particular variant in question, which is why we refer to it as a variant level calibration. And one of the main things that we're hoping to discuss with this group is thinking about how to adapt MAVEvidence for calibration within the oncogenicity framework so that it can be productively applied within the context of doing somatic variant interpretation. Just as an example, we did our own test. So this is not a test of reclassification, which requires knowing what all the other lines of evidence are for a particular variant. What this is an evaluation of what type of evidence can we apply to, for example, BRCA1 VUS within ClinVar. And so at the time we did this analysis, there were 1,360 BRCA1 VUS in ClinVar, and we found that we can apply within using MAVEvidence a range of different evidence strengths to about half of all VUS within ClinVar for BRCA1. Looking a little bit ahead, a lot of times when we talk to people, there's a concern about this kind of data only being available for a small number of genes. And the community has a wonderful resource known as MAVEregistry, which allows people to pre-register studies that they're doing so that they can coordinate and hopefully not step on each other's toes when doing these studies. And if you look into what studies have already been done and what studies are coming in the next few years, you see that there's a high percentage, more or less across the board for these cancer panels, a high percentage of the genes that are included in the cancer panels are covered by one or more of these MAVE studies. And this is not surprising because the community is consciously, emphasizing the most clinically actionable genes. And so, we expect in short order that we'll have more or less full coverage, and that in the meantime, for those most important genes, there will be multiple coverage where you have multiple high throughput assays covering the same variants within the same genes. This is a snapshot of the new version of the application, just to kind of give you an idea of what the user interface looks like. So within the application, you are able to select the gene that you're interested in and search for what variant you're interested in. And the landing page shows what high throughput assays are available for that particular gene. And so here on the screen, we have BRCA1. And there are 6 different assays that are curated for BRCA1. BRCA1 is a little bit of an outlier in that it's a particularly large gene, and so unlike many of the other proteins in the platform, it doesn't have complete coverage across the entire coding region. But instead, these assays have focused on known important functional domains like the ring and BRCT1 and 2 domain. After you search for the variant that you're interested in, what you get back is a report that summarizes what the available functional evidence has to say about your variant. And so here we're looking at the M18L variant of BRCA1, and it happens to be located at the beginning of the ring domain. And right at the top, for people who need a takeaway message, we provide a functional evidence code, again, within published standards, in this case, the ACMG framework. As you can see in the summary section, with the high throughput nature of these assays, it's common to have multiple coverage for a variant. So this particular variant happens to be covered 5 times by 4 out of the 6 assays that were shown on the landing page. And what we do is to take every piece of data and using those clinical labels, produce a corresponding calibrated piece of evidence. And so in this case, there are several strong pieces of evidence, as well as 2 supporting and 1 indeterminate piece of evidence. We also provide on the report a summary of the biological function of the gene that focuses on the aspects of the protein's biology that are being assayed. And then whenever you see a superscript there, whether it's a number or letter, that refers to a notes and references section where people can get more information about how the data was obtained, how it was transformed, and so forth at the bottom of the report. The bulk of the report is made up of these evidence panels. And within each evidence panel, we cite the study where the data was derived from and link out to it. So that's, that green text is a link directly to the study. We also provide some context that gives a brief description of how the assay was performed, what model system was used, what was assayed. And then we always provide 2 different representations of the so-called odds of pathogenicity. One is the one that's directly calculated by the platform, which is the numerical representation. And then we translate that again, according to published standards, into a discrete functional evidence code in the ACMG framework, with the idea that that's how most people will be utilizing this data in practice. And then the last piece of every evidence panel is this so-called variant effect score histogram. So I mentioned that when you cover so many variants in general, a subset of them will be covered by existing clinical annotations. And this is showing how the score of the variant that we're looking at on this report, which is shown in the drop vertical, where it sits relative to the histograms of the scores for the benign and pathogenic existing clinical annotations. So the fact that this is getting BS3 strong recommendation is a result of the fact that overall this assay does a good job of separating benign and pathogenic annotations, and also this particular score is in the bulk of the benign distribution. So if it happened to be the case that we were looking at a variant that was kind of at the center of between these distributions, even for this strong assay, that could receive anything down to indeterminate level of evidence. This is just another evidence panel from that same report to give you an idea of what a different, in this case, slightly older assay looks like in that same representation. So again, we have the link out in addition, and the context and the odds path, which are nearly neutral in this case, because there is this heavy overlap between the scores for the existing annotations, and also this variant is located close to the crossover point between the reference sets. I won't go through the TP53 report in as much detail, but I want to show it just to illustrate that where specialized recommendations exist for handling functional evidence, we are following, for example, the ClinGen TP53 VCEP specifications, which for good reason has particular ways that you combine the functional evidence to come up with an overall recommendation. And so for this particular variant, the overall recommendation is pathogenic strong. And here's an example of what one of the assays looks like in the evidence panel for that variant. Okay, I've been way too much talking, but we did just want to say that we are really looking forward to interacting with the academic nonprofit community. Obviously, understand the importance, and would love to have beta testers on the platform, no fee for both clinical and research use, and also happy to provide multiple seats for the organization so that people who are interested can try it out. And Beth is free to distribute all of our contact information to everybody here on the call, of course, and then we have this particular email address that you can always send emails to if you want to contact us. And I'll just put these discussion points up on the screen. Of course, happy to take questions about anything we discussed in the presentation, but some questions that were on our minds is, what are the tools and strategies that this community is using to search and evaluate functional data, in particular to determine oncogenicity? Does it make sense to apply a sliding scale like we're doing for ACMG on the basis of those subsequent elaborations that have been published? What are the criteria, appropriate criteria for an assay to inform oncogenicity? And similarly, what's an ideal reference set of annotations that could be used to benchmark assays for their ability to tell us something about oncogenicity? That's all we have, I think.
Awesome. Thank you so much. That was really a nice presentation. It's really nice to see a lot of the standards that are built, that have been very recently built, being used in a platform like this. So they're really cool. So are there any questions from the audience right now?
Just let me know if you want me to flip back to any part of the presentation too.
Sure. So yeah, I was kind of wondering, like, how do you deal with genes that might be an oncogene in one tumor type or a tumor suppressor in another one? How do you go about assessing oncogenicity in those cases for those types of genes and variants?
Yeah, I think at a very high level, I think the answer is that so far we have been focused on kind of germline classification,
Hmm.
and it's these kinds of questions that we're looking to get feedback on. But I don't know if Daniel has also spent a lot of time thinking about this. I don't know if you have anything to add to that, Dan.
No, I think that's right. I think that's that's something we're working out now, and very interested to get feedback on.
Hmm. I know from personal experience, like for evaluating functional data, a lot of the time it's doing a literature search or looking at things like ClinVar, HGMD, to figure out, what has been seen in germline literature, what's been seen in somatic literature. I know this group in particular is closely connected with the ClinGen somatic working group and with the civic database community. And those groups have been working together to provide a knowledge base that's based on, primary literature evidence, whether it's evidence giving us data for looking for therapeutic relevance or prognostic diagnostic relevance. But really that's just the tip of the iceberg. I think there's a lot of different like commercial and publicly available entities that are represented in this group too. Those are just the ones that are at the top of my mind.
One thing we have heard from some people, and be interesting to get your feedback on this, is that it's not a full solution, but what we have heard people say is that starting by looking at the available germline classification is a place to start when thinking about oncogenicity. And so some people that we discussed and showed a demo to said that they would go to this as a a way of first starting to understand and then applying their sort of contextual knowledge to refine their thoughts about oncogenicity given the kind of the germline classification.
Yeah, absolutely. And a lot of the tumor suppressor genes that we know of also have like a tumor predisposition when seen in hereditary context. So that can be helpful for us too. I see that Wes has his hand raised. So go ahead, Wes.
Hey,
Wes.
thanks for your presentation. It's really exciting to see more people kind of get excited about MAVE data. Our group closely works a lot with the AVE Alliance also, and I think I've already talked with Daniel briefly on email about how we're, at least I'm going to be at the MAVE workshop in London in July with you guys. So it'll be exciting. I guess some like high level questions for your platform, since with the VICC, we're a lot about federated and kind of open sharing of data. What are your long-term plans for, like development of API for like research use, and kind of like what are you thinking for your platform and how you're going to like utilize then community feedback, and then support like the open community as well moving forward to be able to utilize, this MAVE data and in a way that will allow for like the development of, because a lot of the questions that you have are very ongoing on how can you utilize this data to actually determine like oncogenicity versus pathogenicity. And I think there's just a lot of, as you've probably ascertained already through your conversations, there's not a lot of clarity on how you can utilize and like be really certain on, what this data is kind of providing. And so I guess that was a very tangential question, but what are like the long-term kind of plans for supporting and helping the research community be able to utilize this data?
Yeah. So I should say that, we've been in touch with Alan Rubin and others who are involved in the MAVEDB project, right? Which is, we sometimes get the question when we're talking to researchers, even researchers who are producing, what's essentially MAVE data. Maybe they're calling it deep mutational scanning or something else, but it's, it's MAVE data. And still not yet everyone is aware of, MAVEDB as a resource. And so when they ask us, like, where can we put the data such that it will be, publicly available to everyone and be a kind of source of record for people citing and utilizing the data, we always point them to MAVEDB, because I think like that is what we hope will become the sort of the record for publicly sharing this kind of data. I think, for our platform, what we're hoping is that it'll be complementary to that, in that, it does this sort of these extra pieces that allow it, to be easy for variant scientists, for example, who are working at a commercial lab to apply that data in the context of a variant interpretation workflow. And, I think we have this talk and then several more talks with ClinGen, groups coming up in an effort to really sort of engage the community to figure out what's the best way to continue to interact. And we will have an API available because that's actually also something that, our potential customers are talking about based on their need to integrate this kind of data into their own systems. And so, it's not, it's not available right now. And so like if you've got an evaluation, which we hope people will take us up on that offer, we wouldn't have the API access available right now, but we do plan to support it going forward, because like we said, it'll be necessary for our business in any case.
Yeah, that's great. I guess another question for you, because you talked a lot about also kind of hooking in, other potential studies from like PubMed, just set from a perspective of kind of how we're trying to, because I think I've been thinking about this question a lot too, also with our clinical team. What ways have you guys thought about kind of utilizing data from, the literature to supplement and kind of reassure or detract from the data that's actually being seen like within MAVEDB, right? Because depending on, humans are very complex beings, right? And sometimes, I love the whole point of like what a MAVE is. And I think there's a ton of information to be gained, but at times, some of the questions you've already heard from Beth is like, okay, we are these complex individuals, and how does this work in isolation versus like in an organism? So what ways have you thought about kind of like supplementing and potentially like downgrading / upgrading code assignments based off of like other literature and functional studies?
Okay, I just want to make sure that, I wasn't too unclear. We are, of course, like deriving, all of this data currently from the available literature. So like in general, our workflow is to, identify a functional study, mostly high throughput functional studies, and, to curate that data from the literature so that the raw functional data is coming from the literature. I think maybe you're meaning like other types of data, like clinical data, for example, that could inform how to interpret the functional data.
Correct. Right. Yeah. So like for the ACMG and for the ACMG guidelines and the oncogenicity guidelines, you have functional studies, but you also, that's both in vivo and in vitro, right? So how are you utilizing both to kind of, go ahead and take, because what I think was really interesting about MAVES, right, is it provides you with this high throughput assay where you can see all of this and you're applying some kind of selective pressure, and that's awesome. But then it's also really nice to then actually like see that in like a human study, right? And how can you supplement that to potentially tweak like the score that you end up giving based off of that? Or are you not really concerned about that at all?
Okay, yeah, so what we are already doing is, the way that we calibrate, evaluate the assays and calibrate the evidence are from clinical annotations. And so those are, annotations based on, variants that have been seen in humans and determined to be either pathogenic or benign on the basis of other kinds of evidence. And so that is the data that is being used to both evaluate the assays and calibrate them already. But I think, to your point, there is a lot more we could do. We haven't gotten to the level of considering, specific tissue types and that level of detail. What we're sort of trying to do is, at the beginning, pick the so-called low-hanging fruit, and but still also, apply the data in a way that's consistent with guidelines and is, responsible ultimately.
Yeah, great. Yeah, I mean, it's a massive undertaking, so good stuff.
For sure.
Yeah, and I didn't get a chance to say that we wanted to thank Wes also for presenting the last presentation, because that was a really nice setup and also a good chance for us to see kind of like what these discussions are like.
Thanks. Manuela, I see your hand is raised.
Yeah, this discussion is really interesting, and I want to dig a bit further in your scheme of actually harmonizing your functional evidence. Did I understand correctly that you're basically using available annotation to define your score of functional, like pathogenicity in a sense? Okay, so then the question would be, did you actually benchmark that? Maybe use just like 2/3 of the data and then see whether you could actually fit the other part? You have any information on that?
Exactly. We didn't go into the details here, but yeah, it is important to do that. So, for example, we try to stick to, as closely to the guidelines as possible, but there are gaps in the guidelines, again, because of the evolving nature of these technologies. And so there are some decisions that we have to make in order to do the evidence calibration. And so, where those are necessary, we're applying kind of standard principles like, for example, like doing cross-validation to see whether the level of evidence you get is consistent with the annotations that are applied when they're withheld from the evidence calibration set and things like that. Yeah, because I think everybody on this call realized that, but when you talk to people, other people, it's not always obvious that there's danger in both directions of being both too confident and underconfident when you're applying data for variant interpretation. And so, we do our best, like I said, by applying the guidelines and also standard practices for doing the evidence calibration.
Okay, yeah, that's cool to hear. And maybe another question on your way of setting up or finding actual functional assays. You said you're using PubMed, and so I'm curious, how automatized are you doing that, or is that really a lot of digging into the papers?
Yeah, it's I would say it's like partially automated at this point. So we have, again, thanks to community resources and our own efforts, a good set of like a training set, if you want to call it that, of publications. And of course, PubMed does make all of their abstracts available programmatically. And so that helps in identifying these kinds of functional studies. But every aspect of this at this point is like, in process of being evolved to a more kind of automated and refined state. So, we do look at a lot of paper abstracts and titles in order to identify the relevant studies.
Hey, cool, thanks.
Yeah, thank you.
Great. Kind of just building on that, what are your methods for harmonizing that data that you're mining from there? We've seen in this group's practice that harmonization can be quite tedious, but I'm kind of wondering what kind of tools you've been using for that.
Yeah, so I'm thinking about like, which aspects need to be harmonized. So obviously for clinical annotations, we rely a lot on, what's available in ClinVar and through NCBI. And then for the functional studies, it is more heterogeneous. And so basically like in that scheme that I drew previously, where like on the top, the functional data was coming in and on the bottom, the clinical data is coming in. There's a lot more bespoke code on that upper branch that has to do with bringing in the functional data. So we're trying to be efficient about it and to eliminate the amount of work we have to do for each new functional study. But again, inevitably, and especially given the fact that not everybody yet is knowing to put their data into MAVEDB, there is some extra code that needs to be written for every functional study to make sure that it maps onto, for example, the transcript that we're using as a reference, and is actually evidence on the variant that we're saying it's evidence on.
Cool. Yeah. Thanks.
I think
did,
kind of, sorry, Beth,
go
to
ahead.
build off that real quick. I think, because you're asking about from an oncogenicity perspective now, I think what you'll see, which is something that we deal a lot with my group specifically works on standardization and harmonization a lot. There's a lot of what we call categorical variation in the cancer space, right? So it's not going to be necessarily as clean as like a specific change. It would be like this more broad kind of class. And so it'll be really interesting to see how, you continue to work on like harmonizing data that comes maybe out of like Civic or something that's coming from a categorical perspective instead of like a very specific, germline variant.
Yeah, that's great. I mean, we'll probably reach out to sort of learn more about what you've experienced there as we dig deeper in here.
Just to clarify, is MAVEDB a crowdsourced database?
Yeah, it's totally open source and supported by folks at the WeHi Institute. It's Alan Rubin, and then also at the University of Washington, where it originated. So that's the right place for experimentalists to deposit their data from these high throughput functional studies.
So are you then moving new functional data from publications in there as well, or at least hint to the authors? How's that going?
Yeah, currently we are having to both, pull the data from publications directly because it's, not always being deposited in MAVEDB, and tell people well, I shouldn't say tell, encourage people to deposit data into MAVEDB so that becomes a standard.
But you're not depositing data yourself into MAVEDB if you find them in publications?
Oh, interesting. Yeah, no, we haven't yet, but we should talk to Alan about that.
Yeah, I mean, would be a great starting point for people to see that the database is expanding if you already have additional data coming in.
Yeah, it's a great point.
Excellent. Great. Are there any other questions or comments from the team or from presenters?
No, I guess, I'll just repeat the offer to, just please do reach out if you're, if you have more you want to discuss about these topics here on the screen, anything we discussed today, or also, want to try out the platform. It's totally free. We've tried to make the setup process very easy. So happy to, interact more with people from this community, especially.
Fantastic. Thank you so much. We're almost at the top of the hour, so thank you again for coming and presenting. obviously this topic is really interesting to everybody here. And Adrian, I will send out the email address along with the link to the recording to the VICC distribution list. So I think we should be good to go. So if you guys are okay with me sending out your email addresses to the team, I will do that later today along with the recording. So thank you for being here. This is a topic of great interest, and can't wait to see what comes with all of your work and collaborations to come.
Great. Thanks so much. Thanks, everybody.
You bet.
Thank you.
Thanks, everyone.
Thank you.